Hello, folks. Here we are at Unit 6, DNA and Protein Synthesis Review. So we're going to move right on down to where we have our multiple choice questions. Here we are. Which of the following is an example of a genetically engineered organism? Well, if you've genetically engineered them, you're changing their genetics. You're changing their genes. So let's look at our answers. We have a plant that received external DNA to produce natural insecticides. Well, I would say that's changing your genes. B says a seedless fruits resulting from grafting. Now, grafting is putting two plants together. That's not actually changing their genes, just how they grow. A new plant variety created by cross-pollination. So again, that's not engineering them. Cross-pollination would have to be a natural process. Humans could do it artificially themselves, but you're still not necessarily genetically engineering them like in a lab. All right, and then D, a plant that naturally possesses them. Well, that's not changing their genes at all. They naturally possess it. So that should be A. When you are receiving external DNA from another source, that creates recombinant DNA where you recombined it. Number 2, look at our DNA segment over here, which would go with it. So hopefully you know the rules that apples go in the tree and cars in the garage. So if this is an A, the opposite side should have to have a T at the top. So we have 2 that have T at the top. And then the second letter is a C, and the cars go in the garage. So that should be a G with it. Now that one no longer works because it has an A. This one has a G, and then just to check, G goes to C, T goes to A, so you should have gotten D for that. Here's using our mRNA chart with our 3 letters. Remember, first base, second base, third base. So if we have A, G, C, you start with the A over here, and you're somewhere in that row. Then you have G coming down, and so they come down, you're somewhere in these 4 right here. And then C tells you it's the second one down, so you should have gotten B, sir. Complementary base pairs linked together. What they're linking together, your bases, obviously, are your nitrogen bases. It has it right in there. So that should have been C, nitrogen bases. Number 5, which scientist discovered that DNA from any organism has the same amount of guanine as cytosine? Well, that was Chargaff. So Watson and Crick discovered the structure of DNA. Mendel's a genetics guy. He wasn't even the DNA guy. All right, so Chargaff said there were equal numbers of C's to G's and A's to T. Number 6, which scientist is credited for developing the first picture of DNA using those X-rays? And she got no credit for it and actually died for her work. That was Rosalind Franklin. Number 7, after a culture of cells is allowed to multiply, the cells allowed to multiply and looked at through a microscope, the cells are X-rayed with high energy radiation for 1/100th of a second. So you're exposing them to radiation. After radiation, many newly reproduced cells appear different. What's likely occurred? Well, if you expose cells to radiation, they look different afterwards. You probably got a C, mutation. Number 8, a scientist discovers that the strand of DNA he is using is 20% guanine and 20% cytosine, which makes sense because they should be equal. They go together. So that means we've already used 40% of our strand. So only 60% is left, and that 60% has to give us both our A's and T's, which Chargaff said have to be equal. So if you have 60% and you've got to have equal amounts, then you're going to have 30 of each. So you should have B, 30% and 30%. Number 9, what kind of bond is depicted there? So you need to know the kind of bond that is between your bases. So A and T has 2 of these bonds, C and G have 3. Those are your weak C hydrogen bonds. All right. Peptide bonds are found in proteins. Covalent bonds are actually between the sugars and the phosphates on the side because they're strong bonds. All right. And the phosphate bond is not even a thing. Not for this. All right, number 10, the 2 molecules that form the backbone of DNA, well, we just saw a picture of it. What makes the sides of our DNA are the phosphate and the sugars because the bases are in the middle making the runs, okay? Identify the correct transcriptions of DNA to mRNA. So in this, you need to remember that mRNA has a difference from DNA. Instead of T's, it has U's. So right away, You got B has T's in it and D has T's in it. Neither one of those could be your answer. You don't even need to look at them. So now you're looking at this. So does A go to U? Yeah. G goes to C. C goes to G. T goes to A. This all makes sense. Let's look at this other one. C goes to G. That's true. T goes to A. That's true. G goes to C. That's true. A to U. Well, it says all the correct ones. So it looks like there's more than one here. And so both A and C are correct, and the ones with T is obviously incorrect. All right, number 12, what could replace B? So these are the processes involved in protein synthesis. And so going from DNA to mRNA, right, where you're copying that code is transcription. They've transcribed the directions. But going from mRNA to a protein, you've completely changed the language. You're no longer in a nucleic acid, you're in a protein. And so now you've done translation. So transcription first, then translation. They make sense to what the words say. Also, they go in alphabetical order. So the C in transcription comes before the L in translation. How many amino acids would be linked to form a protein based on this? So how many letters, basically, do you need to use in your chart? Hopefully you know 3. So sets of 3. 1, 2, 3, 4. Okay? Should be 4 amino acids. That's why I always made you space on those worksheets we did to keep them separate. This makes it easier to see. 14, select all the statements that are true about DNA transcription. So making a copy of DNA. Both strands of DNA are copied to form 2 strands of mRNA? No, only 1 strand. Transcription begins with the unwinding of one section of DNA nucleotides necessary for the production of one protein. Well, sure, you got to unwind your DNA to make that copy. That makes sense. So B is correct. C, one strand of DNA acts as the template. Yeah, that's correct. There's 2 of them that are correct. The newly formed mRNA strand is synthesized into fragments that are chained together in the cytoplasm. No, we're not cutting up into little fragments. No. Kind of. All right, 15. During translation, free-floating amino acids in the cytoplasm bind to specific tRNAs because they're transfer RNAs. They're transferring the amino acids. What are the further steps of translation? All right, so tRNAs have picked up amino acids. What else has to happen? A, mRNA and amino acids travel to the nucleus. Well, where does translation occur? Is that in the nucleus? You're making a protein. That's at the ribosome. So A is incorrect. tRNA supplies the amino acids to the growing peptide bond. Yeah, that's going to happen. They're going to bring those in there. So B is correct. C, tRNA binds with tRNA? No, that's not what happens. D, tRNA is associated with a specific amino acid matches to the mRNA. Yes, that occurs. All right, 16. Each triplet of nucleotides in mRNA that specifies a particular amino acid is called. So 3 letters on the mRNA is called a codon. 3 letters on your tRNA is anticodon because there's a T in anticodon. Zon I made up, and tRNA is just another RNA. All right, number 17, an mRNA strand has the following set of nucleotides. A teacher is asked for students to organize the strand into sets of codons. So how many letters are in a codon? Same number of letters you use in your chart. You got to separate them into sets of 3. So that should see B, where they're all set in sets of 3, the codons. All right, in eukaryotic cells, mRNA travels from the blank to the cytoplasm during transcription. Well, you have to know where everything starts. Well, if you're doing transcription, this all starts with DNA. And where's your DNA? B, in the nucleus. All right, guys, hope that helps. Bye.